Welcome everyone to the ACTnet Pharmacy TV research webinar. We've got a wonderful lineup today. We've got two pharmacist TB researchers who'll be presenting on their research and exciting things that they're doing. And we've got two Queensland-based chairs who are aptly dressed in maroon. So I'm Amy Legg. I'm an infectious diseases clinical pharmacist, and I completed my PhD in Staph aureus bacteremia. So I'm a bit to the side of the TB world. And actually, Dan, did you want to introduce yourself?
Yeah, sure. I'm Dan Hicks. I'm a senior research pharmacist and project manager with the Format Trial, working in Brisbane here in Queensland. But I'm a pharmacist by trade.
Yeah, that's great. I'm really happy to have Dan with me helping out this afternoon. We're also very lucky to have Anton, who's an editor at Clinical Pharmacokinetics, and he'll give us a little introduction to that later on today. I did want to acknowledge that Dr. Burke was planned to chair this session. However, he's currently on extended family leave. Importantly, in Australia, we always acknowledge the traditional owners of the land where we're meeting. So I'm in Meanjin in Brisbane, and I'd like to pay my respects to the Turrbal and Yagara people and their elders and past and present. And I would really like to extend my respects and gratitude to any Aboriginal and Torres Strait Islanders that we have in the session today. But I think that's enough of the formalities, and we have got a few extra people, which is wonderful. And so I'd like to introduce our first speaker. So we're very lucky to have Dr. Shukry Zawahir, who is a research fellow at the University of Sydney and has expertise in tuberculosis, antimicrobial resistance, and global health. He holds a PhD in pharmacy and has extensive experience across Australia and in the Asia Pacific region, including multinational TB trials and community-led TB initiatives focused on improving care in low and middle income countries. And so we look forward to your presentation today.
Morning and good afternoon, everyone. Thank you for kind introduction. As Anne said, I'm a pharmacist as a profession, and I started with the pharmacy work a long time back, and then later on, I will go through my profile later to TB related studies. I'm very happy today to share my research with you all. Right. So this is my academic profile. I did pharmacy about 2 decades ago. And then immediately my pharmacy degree, I started working as a clinical trial coordinator. We were conducting a huge clinical trials in Southeast Asian region, phase 1, phase 2 clinical trials that is more on toxicology related clinical trials. And then I did my pharmacy, social administrative pharmacy degree as a master at University of Science Malaysia in Malaysia, Penang, where I also did sort of work as a research officer conducting some sort of smoking cessation studies that is also multinational studies. Then later I have completed my PhD at University of Sydney, Australia in 2020. And then I started working on AMR related projects in Sri Lanka, Australia, Vietnam, Indonesia, and also some TB stuff. We will discuss in detail also the TB related research what we have been doing in the region. And on my current work, yes, I've been continuing with AMR TB related research in Australia and the region. So it's a kind of my experience and my training a bit not like this a flow, but it's a different area, but it helps me a lot. So in Malaysia, in terms of my profession, after completion of my master, I was a lecturer in pharmacy, and then I completed 2 years of full-time lecture in Malaysia, at one of the university called Management Science University in Shah Alam. Then later, I was a senior lecturer at the University of Rohuna again, pharmacy senior lecturer when I had a chance to work there for about 2 years. And then I joined University of Sydney for my PhD. Since then, I've been working as a PhD student, and then since 2020, I've been working as a research fellow, postdoc researcher, and a research fellow currently. So what I have learned so far, like teaching helps me to communicate clearly my research goals and research expectations. And then also throughout my career, last I can say 20 years, I have done a lot in different countries in terms of conducting different types of research. If you have any question, I'm very happy. You can interrupt me and ask, and I can discuss with you if you have any exciting questions. Right. So in my PhD journey at the University of Sydney, uh, I started with actually AMR-related study in Sri Lanka, in which I try to understand inappropriate use of especially antibiotics in the community settings, also community pharmacy setting in Sri Lanka. And also based on those formative studies, we developed kind of intervention program in order to, I can say, an antimicrobial stewardship program, especially for community pharmacists and community, in order to improve appropriate use of antimicrobial. So during my PhD, I able to publish few papers, but when I was in second year, I published some papers that is more on simulated client or standardized patient survey, which we plan or we develop some sort of study design, where public or trained to act as a simulated patients, and they visited pharmacies and reporting the symptoms of, especially we were focusing at that time, minor ailments, something like, viral upper respiratory tract infection, symptoms of viral upper respiratory tract infections, and also UTI and other infections, and see actual behavior of pharmacy in the pharmacy. Also, we were doing some kind of cross-sectional survey among countrywide cross-section surveys among pharmacists and public in order to understand the, the, their knowledge and their attitudes and behavior related to antimicrobial use in order to develop some kind of intervention program. So by second year of my PhD, I managed to publish 2 papers, and one of those are the simulated case study. Then I was invited to be a part of another study group, which they receive a DFAT funding to do some kind of similar studies in Vietnam to understand public and also pharmacists and policymakers and healthcare professionals behavior in relation to antimicrobial prescribing, dispensing, and use. So it was a good opportunity for me because of my publication. It was attracted. So especially in the region. So then we were like collaborating with Indonesian, and also the similar thing we doing an Indonesian team, and then with we were like from Australia with the Vietnam team. We were conducting kind of formative study again to understand the problem related to AMR. So one of those scenarios or simulated patient cases was reporting symptoms of TB to the pharmacy and then see what's their behavior, something like that. So that is my turning point towards focusing on TB related research in addition to MR research related research. So what I can say, like though I'm a pharmacy practice of a clinical pharmacy related researcher, my publication credibility and pathway into AMR TB and then implemented research are like a very kind of focus. So from the main key point towards TB research is my studies being conducted in Vietnam. So I will have some detail about those studies which we were doing in Vietnam so that you can understand. So based on the evidence from my PhD and publication, I started working on a huge project that funded by the Fed, that's AMR resist project, and then we came up with some gaps in TB, and then we developed some interventions. So I will discuss those things shortly. So key shift is like describing pharmacy practice problem and designing practical solution with the partners. That is, what I learned from my career in terms of pharmacist researcher and TB researcher later on. So especially my publication in my higher studies or HDR study, it's evidence for collaboration and funding and implementation, TB-related implementation studies. So as I said earlier, so we were working in the community and community pharmacies in 3 different countries like Sri Lanka, Vietnam, and Indonesia to understand antibiotic supply for common infections and public antibiotic use. And also we conducted some standardized patient surveys in all these countries. So this gave us a very clear picture how the community pharmacies in this region respond to viral cough or viral respiratory tract symptoms, upper respiratory tract symptoms, and bacteria infections. Then I would like to highlight the very important study we were doing in this region, like standard patient survey among pharmacy or pharmacists. It's a method of understanding the actual behavior of the pharmacists when they encounter with the patients. So we develop scenarios, train the actors, and then ask them to visit the pharmacies and the standardized way. And then we have some kind of checklist or questionnaires, and then we develop some evidence how actual behavior in terms of responding to symptoms of TB, upper respiratory tract infections, viral infection, viral diarrhea, so on and so forth. So this is a very alarming findings from our mystery shopper or standardized patient surveys. Like up to 61% of the pharmacy gave out antibiotics for overall common cold and acute diarrhea, kind of viral infection in Sri Lanka. And these were published. So this publication led to work in Vietnam, one of the big projects funded by the Fed. So in Vietnam, we did the same sort of standardized patient survey in addition to many other sub-studies or projects we did. So I'm just highlighting only one. It was a huge 4 years project we did in Vietnam, and also now we are continuing as a One Health projects afterward. Right. So in terms of common cold presentation to the pharmacies, like 92% of the pharmacies were giving out antibiotics for viral common cold symptoms. Acute diarrhea, about half of the patients or 50% of the pharmacies, and then presumptive TB symptoms like TB symptoms like a fever, cough more than 2, loss of body weight or unexplained body weight changes, and night sweat, something like that. Patients or standardized patients were reporting, and 90% of the pharmacies were giving out antibiotic for this. And some of the cases, like very strong antibiotic, like fluoroquinolone, were given. And also presumptive MBR-TB symptoms were presented again. Up to 70% of the pharmacies were giving out antibiotics. Were published as well in the Lancet Regular Health, also given JPLOG. So if you look at my right-hand side, there's some facts. So interestingly, when they were presenting these viral symptoms, so the pre-prepared packs were given out mostly for 2 days, like different colors of medicine without names and all. So these medications were given out to the patients or standardized patients for their viral cough, and we identify some of them were antibiotics. So most of the pack had antibiotic, and alarmingly, these were given for only 2 days, including TB, presumptive MDR TB symptoms. So this study, though we were trying to understand appropriate use of antimicrobial, this presumptive TB antimicrobial use for 2-3 days especially strong antibiotics, something like fluoroquinolone. It was very alarming for us. So that is the turning point. In addition to antimicrobial stewardship program, we were planning. We also received some seed funding, and then we have started developing some intervention programs, especially for pharmacists, in order to identify symptoms of TB and then also appropriately refer these presumptive TB or MTR presumptive TB patients to a TB clinic or hospital for TB screening. As you know, TB is a huge problem, especially in Vietnam, Indonesia, and the region. Like 1 in 3 people with a TB are never diagnosed and not receiving inadequate treatment in the private sectors. And according to literature, 40 to 60% of TB patients visit pharmacies before accessing TB services. So it's a huge number of patients in this region. They go to pharmacy for some help with the TB symptom. So in our study, only 5% of the pharmacies with a presumptive TB, the patients with the standardized patient with a presumptive TB were refer these services for TB screening, and 90% were given out antibiotic. So that was alarming. So then we understood, based on the literature, based on our finding about TB, pharmacist behavior in terms of TB symptom, we understood it was a huge gap. So we have developed an intervention program with some WHO guideline, and WHO and FIP combinedly developed some infographic materials and also some training and so on and so forth. And then we have just started study in Vietnam, and we are going to study a pilot version of the same study in Indonesia soon. Purpose of this study is symptom-based. Training the community pharmacists in order to identify the TB cases based on reported symptoms, and then we want symptom-based screening, and we were training them to refer these patients to a hospital or clinic in order to test for the TB, chest X-ray, and then the bacteriologically information test, and then TB treatment initiation at the hospitals. So this is my one point turning point towards working on TB studies. So in these 2 countries, in order to identify TB cases through community pharmacies, we collaborated with the universities in Indonesia and Vietnam, and pharmacies, pharmacy associations, and clinics, and national TB programs in these countries, and working together in order to establish very cost-effective way of TB case finding through community pharmacies. And my second area of TB research is we have been doing TB preventive trial or phase 3 clinical trial. I've been managing this trial in Vietnam and in Australia, 3 sites in Australia, 2 sites in Vietnam, and in collaboration with Canada, US, and other countries funded by TB's US CDC. This has started 2021 and it's ongoing until 2030. The intention of this trial is to reduce the TB preventive therapy or TPT from standard of care to 6 weeks of treatment. So reducing from maybe 4 months or 12 weeks to 6 weeks. So our sample size is like the 3,000 plus patients. We have recruited around half of those patients so far. It's a multi-center trust randomized control trial. Not blinded. And then we have another phase 2 trial. We just started, same funded by the US CDC in Vietnam. So I have also involved in this trial. So this is phase 2 trial, reducing, we call it CRUSH, TB treatment trial. This is a kind of, many different regimen together. We are trying to reduce the TB pulmonary TB treatment from whatever 6 months to 12 months to 8 weeks. And as a sub-study, we have started again together with other investigators in other countries, quality of life of these TB patients or TB infection patients who are receiving TB treatment. So we were assessing quality of life. So that I got, so I've been working in these trials since 2021, in addition to that TB case finding through community pharmacy. And so through the TB case finding community pharmacists, we are kind of helping in improving the cascade of TB care. And also in addition to that, AMR related works, we are doing kind of AI-driven drug discovery and TB preventive qualitative life, as I said, asteroid trial, and also prescribing appropriate. One of my PhD students is doing, in order to improve the quality of management of the elderly patients, a tool develop and then validate the tool in order to improve the geriatric patient care. And also we do crush trial, and so based on our baseline studies of AMR related in Vietnam and Sri Lanka, and also in Indonesia, now we have developed the antimicrobial stewardship program for community pharmacies at Gamiji. We have done the pilot work, and also we have established one of our team's PhD student has completed her PhD in order to develop AMS programs of 40 hospitals in Vietnam. And then next study we have developed, and we submitted an application for AI-enabled cough triage study, which is helping community pharmacists in order to identify the cases using trained AI model. That is very exciting one. It is in a multinational study. We have submitted e-Asia grant, submitted as a PI in Vietnam, Indonesia, and Philippines. And also the other study is a patient-centered implementation study, which we are planning to submit and grant application near soon. So key lesson I learned so far, like in my research career, started with the MR and antimicrobials, and then TB. Like start with the practice. So based on the practice of behavior, we understand the research questions, and then publish early. We'll have some very good opportunities to contribute, and also build the partnership in the region or wherever you work, and or of course accept the rejections. Always try to be persistent, because rejections are always there. So if you are persistent, we can get through those with rejection. And of course, we need to start with a small project so for early career researchers in order to establish ourselves. I'm also still an early career researcher. And in terms of TB opportunities, I can say like we have TB case finding studies at the support and medication safety, mentorship, and implementation science and clinical trials we've been doing in terms of TB. And so these are the few challenges we pharmacists face. We always think of like limiting our career at dispensing community pharmacists or hospital or clinical pharmacists, but it's beyond that. We can do a lot of research. So we should have a clear starting point, and then also we have, like with the clinical works and other things, we have some time limitation to contribute to research that should be addressed, and limited funding opportunities. Of course, fundings are always very competitive, so we should have you should work towards like getting funding and get through those the struggles. And then we should find appropriate research partners or research team to work with, and also always rejection is there, so we need to be prepared for accepting the rejection. The important thing I found in my career as a TB research, I can say, like importance of pharmacist contribution in TB is still underestimated. So, but as I said from a WHO report and other things, TB case finding through pharmacy can be improved by 60% if we work together. We can help. But the states already started with the WHO program, public private mixed program or something like that. Still, as a pharmacist, we can contribute a lot. And the final some take-home message is pharmacists can contribute beyond the medical supply or clinical work. PDMR research needs real-world pharmacy support because I used to say pharmacists are gatekeepers, especially in low and mid-income settings. So start with the small things, and then we can develop our career. So thank you so much for your kind attention. I'm very happy to have any questions or any suggestions. I'm very happy to answer. Thank you so much.
Thank you, Shukry. What an interesting talk. Your work with the pharmacies and the antimicrobial stewardship is fascinating. I'm curious to know, with the 90%, I think it was 90%, people were prescribed antibiotics inappropriately. What do you think are the contributing factors to that in those environments? What do you think it's like adaptation, or are there like 90% is pretty high? I'm wondering what kind of factors you're up against.
Yeah. So I just highlighted the behavior or the dispensing or prescribing these things. So in addition to that, we had a lot of other sub studies like qualitative interviews with the pharmacists and policymakers and doctors. Also, we did a health system analysis and to understand. And there are a lot of factors contributing based on our other studies, and based on this study. Number one is uh, patients, they think the antibiotics are kind of strong medication that can cure anything. So patient demand is there based on the understanding. So number one. Number two, pharmacies, they believe that their business is important than I mean, if they don't sell antibiotic as patient is expecting, then that may impact negatively for their business, and patient may go to the other pharmacy or some other pharmacy and get the medication or antibiotic they require. And and also we found that patient demand is there from doctors. Also, a quick fix, and then they have some barriers of going to the hospitals, like time consuming and all those things. And then the pharmacies are open long hours, so they can go and access. And alarmingly, one more important things we found, like we did another study, which also the public, that is called mapping study. We mapped the pharmacies across 40 districts in Vietnam, and unfortunately, based on our definition, we could not. We found like around half of these pharmacies were we mapped the pharmacies and we matched the registration detail with the Ministry of Health registration log or record, and we found like the half of them were, like, according to our definition, were not registered as a pharmacy. And also there are some other barriers, like in Sri Lankan studies, we were like trying to identify this availability of pharmacists in the pharmacy during the business hours. That is also lacking. Sometimes the pharmacists registered on the particular given pharmacist name, but most of the time pharmacists may not be there, but someone else run the business without any pharmacy qualification. So these are a lot of challenges. We came up like it's like a multifaceted issue we need to tackle with not only pharmacists, consumers, doctors, policymakers. It's a challenging, complex issue we need to address multifaceted way.
All right. Well, thank you, Shukri. We'll move on to our next speaker, who's Dr. Thi Nguyen, who's a PhD graduate from the pharmacy school at the University of Sydney. Thi's research aims to enable personalized drug dosing in TB and infectious disease, developing saliva-based tests and decision support tools for use even in low-resource settings. Thank you, Thi.
Under Shukry had very interesting points about TB research. I would like to start with a question. Did you know that tuberculosis is the curable, but yes, also the deadliest infectious disease in the world? I didn't know that before my PhD, but yeah, now I know. So think about that. What perspective that gives you about this statement? So for me, it means that we do have effective treatment to treat TB, but the thing is that the treatment is not available where it's needed the most. And do you know where it's needed the most? Based on Shukry's presentation. So it's in low resource countries, even Indonesia and other Pacific Asian regions and African regions. And the thing is that the highest burden of TB happens in the lowest resource setting. So it's all about health inequity. And so I spent my 3 years of PhD trying to improve TB treatment. So it means to improve the access to TB treatment to where it's needed the most. That means low resource settings. So a bit of my background. So as many of you here, also have a pharmacy background. And then I did a master abroad. It's a French program. And then I'm still curious about the world, so that I did a PhD in TB accidentally. Yeah, because in Australia, I had to choose supervisor first and then the topic. So TB chose me. But I'm involved now, and I have to say that I'm not fallen in love with TB, but I'm fallen in hate with it. It's too complicated, and it's like a global challenge that like many practice minds in the world working on that, but it's still ongoing, and it's still the thing. Most people think that it's already the past, but it's not. It's still now. So finishing my PhD, I'm still working on a current project. I went to the Union conference last year. For those who don't know, that is the largest TB conference in the world. And I just feel still so curious, like where we are actually in TB, the roadmap to the end TB goal in 2035. So that I want to, yeah, just like summarize our progress so far with this conference. I will come back in a minute. So maybe we can get started with my PhD project. It's about saliva testing. It's unfamiliar to you because as you may know that TDM for TB is start with a blood test, but who likes blood test? It's very painful, and and then most of the time we need to send it away to the centralized lab through the cold chain logistics, and then requires like very sophisticated LCMS system to analyze samples, and then it needs to come back with the cold chain logistic. Oh no, come back with the admin issue, and then it takes days to weeks before the result available for the clinical team. And it's too complicated, you can imagine, for low resource setting. So that's why we are trying to simplify all of this process into a simpler saliva test, as you can see on the screen. So we hypothesize that we can develop a test, a simpler one that the patient can self-collect it, and then it can be analyzed by a point of care device. So just at the bedside in the community where it's needed the most in low resource country, where we don't have all of the sophisticated laboratory system. And so the two drugs I've been working with, levofloxacin and linezolid. So they are key drugs in the multi-drug resistant TB treatment recommended by the WHO. And they both have high pharmacokinetic variability. That's why TDM is recommended to balance the efficacy and safety. And to make the saliva test a reality, we did a validation study in Vietnam, a low resort country, high burden TB. But you can see it's a beautiful and well cultured country. So let's make our work. So we did a community based cross sectional PK study here. Actually, this is kind of a scaled up study for levofloxacin, including about 60 participants. We did a similar study, smaller one in Tanzania before. By my supervisor, with positive result. That's why we wanna double check here. And this is the first in TB patients for linezolid. So yeah, let's look into it. Yeah, it's quite exciting. Because linezolid is quite an amazing molecule. It can penetrate almost everywhere in the body. So saliva is not an exception. So as you can see in the scheme that we took the blood and saliva, so both of them simultaneously, for 3 time points. So before the dose, 2 hour after dose, and 5 hours after dose. So 3 time points, and then with those sample, of course, we will, for the control purposes, we analyze them using the gold standard method, which is SCMS. So liquid chromatography using the blood. And this is where it becomes interesting. So we compare the result with those analyzed by the point of care test using saliva. So it is a photometer. So here you can see how it's interesting, which is drop in a saliva droplet. Yeah, in the board, and then you can just touch a button, and then the result will be available within seconds. So with this kind of technology, we can differentiate different concentrations of the drug based on the light absorption. So I hope that you are convinced why saliva is cool, because it's non-invasive and it represents a free drug in the body compared with blood. And when it's coupled with a point of care device, the whole process becomes simpler and faster compared with the traditional approaches. And I won't go to technical details today. It's not the purpose of today. Just to show you some promising results from my study. So this for levofloxacin with about 60 participants. So you can see a quite good correlation between plasma drug exposure in using LCMN versus saliva drug exposure using the point of care saliva device. And similarly for linezolid, so this is the very first result in patients with TB. And we also got a good correlation between the two approaches. So I was almost crying looking at this photo, because it's kind of a celebration for patients with TB in low resource setting. And with a larger sample size for levofloxacin, we were able to calculate the ROC curve to demonstrate its diagnostic performance. To figure out the subtherapeutic drug exposure for levofloxacin, and compare with the WHO minimum standard for diagnostics. It actually met the minimum standard, so with more than 70% of sensitivity and specificity. And actually, there's a small portion of the cases that a bit off would form positive results, because it's likely due to the interferences in saliva. There's other stuff other than our drug of interest, like food or other drugs, or interstitial substance, like also floating in saliva, that might interfere with the drug signals. However, is it good enough, when compared with the reality in the field, that there's no TDM survey at all, then no LCMS, nothing at all. So this test might offer something useful to them. And then that's all about my PhD. So this now we're moving to my current TB project. So this kind of a passion one, because many of you in TB know that there's a funding cut last year, and a lot of projects shrinks. But I believe in human potential, that together we can do things. And this disclaimer dawned on me in the union conference from a very famous American journalist, John Green. He said that the real cause of TB is not bacteria, but humans. And although a bit skeptical, I agree with that. Though when you think deeply about that, it's not bacteria, because bacteria, we know how to treat it, but the way we build the system to include some elite ones and exclude others. So it's all about health equity. It's not about science at all, if you look closer into that. That is how my this project was born. So I just have a strong desire to gather all of the insights from the largest TB conference in the world, and then I would like to summarize all of the takeaways and then filter out the key signals from the TB science so far, and then we can build a roadmap for NTB in 2035. That's the goal. But I cannot do that alone. However, we've got AI nowadays, so it's become more and more powerful. So we like to use AI to analyze all of these data and evidence. And I then will build a community. I feel that we still lacking of community engagement in TB. So that I started talking about TB and starting with my LinkedIn. So I think everything starts with a commitment. So yeah, I will start with an individual level first. So somehow we can influence the decision makers in TB research, and then hopefully one day we can also influence the global leaders, to commit to end TB. So I think that's the only way that can definitely end TB. And so in my free time out of work, I will work on this project. So I will, summarize all of the findings so far about TB research. Yeah, everything to end TB to serve millions of people out there. This slide's a bit busy, but I won't go into details. Just to the main idea here is to use at least 3 large language models, so ChatGPT, Gemini, and Claude. And essentially Claude, so that I will extract all of the data and then run the level of confidence and then find out the top signal of the research. And with the goal to end TB. We have talked a lot about achievement, but just to be honest with you, I also have my ups and downs. But one thing, I think what keep us keep going is our philosophy in life. So I think the key things is our purpose and compassion. So built in with a lot of curiosity and courage. And also we need to be consistent, as Shukry mentioned that there's some dark nights that we need to go through. So we need to be consistent to work in TB research. It's a long game. And this is my last slide. So my bottom line for you today is that it's now time to end TB, because if not, TB will end us. And that is true, because I would like to end up with a fact that about 10 million of people are affected by TB nowadays, and about 40% of them still left undiagnosed and untreated. And that is, that will increase the risk of drug-resistant bacteria, and that would be the threat for all of us. So we must take action today. And thank you, everyone. I think the last is my acknowledgement and my contact details. Thank you for your attention.
Obviously, it's absolutely fascinating to think about PDM of these drugs in saliva. Obviously, the drug's got to get into the saliva for you to be able to measure it. And then are you trying to convert it to a blood concentration, or have you, with your RAC curves, essentially developed the saliva targets that you need?
Yeah, it's a very practical questions. Yeah, actually, there's a lot of discussion on this topic. Yeah, honestly, similarly with the dry blood spot, actually, Jan-Willem, my supervisor, yeah, and his team, have been working on this, for dry blood spot. But I think it's kind of applicable to saliva as well. Yeah, as you know that we don't have the therapeutic target yet in saliva, but now so at the moment we need to translate into blood-based target. But it's like the very end. At the end of the day, we would like a saliva target, of course. I think we'll want to comment on that.
Yeah, I wonder if he's got a tricky question. Go, Jan-Willem.
No, I don't have a tricky question. I think that there's two pathways that we can follow. One is that we see the alternative sample as a replacement for the blood sample, and then you would convert it to the blood sample-based target. The alternative would be to do a trial where you validate whatever saliva exposure you measure against the clinical response, clinical outcome, or side effects. So far we have worked on conversion, but at this moment there's a new trial happening in Vietnam, and in that trial we will measure saliva exposure and follow up these patients as part of the trial that's happening. So then we will have actually validated saliva targets.
Oh, that's fantastic. And we do have a question in the chat about how accessible the mass spec would be in low-income countries or the analyzing equipment.
Yeah, I think I can take that one. Or I'm from my country perspective, that the LCMS is not in routine practice. It's only for research purposes at the moment, so we don't have the equipment and also the staffing either. So that's quite challenging at the moment to do TDM in practice. Yeah, so it's like in kind of regional and rural, but of course in the centralized areas they do have the LCMS, but to some extent for the TDM.
And actually, the point of care that you showed, is that something that potentially could travel out to rural areas and be used?
Yeah, sure.
So just a financial outlay there, not needing.
Yeah.
Oh, that's very interesting. And any other drugs? Oh, sorry.
No, I think it comes with a battery pack for 8 hours, so you don't even need power. You can just run it on its own.
Yeah, it's like an electric car. You just need to recharge it.
Oh, that's brilliant. And I might, just before we move on to Anton, I might ask one of the questions that was submitted beforehand. Oh, this might be a little bit. Anyway, see how you go. What do you think the biggest barriers are preventing novel anti-TB therapeutics from successfully translating from early stage research into clinical use? Have you got much thoughts on thinking about sort of the rural and remote settings and that transition of really anything from research into clinical practice?
Maybe I can give my point of view first. I think it's the drug development is very much depends on the interest of industry. When you give them a lot of profits, but TB is kind of the disease of the poor, and no one want to develop something to sell for someone who like cannot afford it. But however, I got to know that GSK is quite on the top of it. Like they are like quite active in TB research, and yeah, I think there's some companies working on that. So the biggest areas to me is funding, but maybe others can have other views on this.
I think you're definitely right. There's a lot of but being able to procure the drugs if they're expensive has traditionally been a big problem, and any drugs that require monitoring, even if it's not necessarily TDM, but any sort of clinical monitoring, then that infrastructure's got to be in place as well. And of course, with any antimicrobial, if it's really good for resistance, we restrict it. Jan-Willem?
Yeah, I think besides the funding, another problem is that TB is a very the disease that takes a long time to cure. So if you compare it to normal bacterial infections, you give 2 weeks of therapy, you follow up for a month, and if they don't relapse, you consider them cured. So for TB, that means you treat them at least for 6 months and then follow them up for a year or longer to see whether they get a relapse. So that makes trials notoriously difficult and expensive because of the long follow-up.
Yeah, not only funding, but more and more funding because of the complexity of the disease.
Yeah, and I just want to add some more like to that. So then that comes to the thing like inappropriate use of TB antibiotics. For example, if we want to treat the patient for 6 months and then patient not to like tend to take the medication as prescribed, then that may lead to MDR TB. Now Vietnam is suffering with a huge crisis with MDR TB and further XDR TB and so on and so forth. And then there's no medication, in the pipeline in order to treat, multi-drug resistant TB and all those things. This is challenging. Also, I agree with that um, the TB is prevalent in low and middle income countries, and then even though if there's a drug trial and then come up with if it is expensive, the affordability and also one issue.
So I'll put all these out to all of the speakers if you want to jump in. So the next question that we received before was, where can hospital pharmacists find research opportunities in TB management?
Yeah, that's the practical one. Yeah, opportunities. I just get when I'm being here, that a lot of job opportunities that are actually available through your networks. It's from the mouth. Actually, like the job by mouth. I just need to ask people in TB research. I think the kind of the decision makers, the research lead, or the manager of a hospital. For example, Jan-Willem is someone here in Sydney, but I think also Victoria also there's some like TB research hub. You just connect to people working over there, and they will and I think that is the ways that I would go for. But maybe Shukri can comment on other opportunities.
Yeah, of course. I can add to what he just mentioned. There are which I know, like there are a couple of clinical trials, phase 2 trials, phase 3 trials, and also many other trials on the pipeline. We are going to not we, I mean, many other groups are going to start in Australia as well. Yeah, especially preventive therapy and also things, also pulmonary TB treatment and all those things. So it's currently happening, and in the future there are so many trials on the pipeline to be started. That is one thing we can start with. Also, in addition to ACTnet and also TBCRA, doing amazing work on TB area. That's like, prevention, treatment, and adherence, quality of life of TB patients. There are so many platforms. The hospital pharmacists, when they are treating the patients with the TB, think of like, so not only just clinical trials, maybe some other adherence studies. Pharmacists always we think of adherence, compliance, and all those things. There is one area, quality of life of these patients and all those.
Thanks, Shukri. Thanks, T. It sounds like you find your niche and find your community of people who are like-minded. I can't see Anton back yet, so we might jump to another question. So the next question is, which medication should be withheld first if suspected drug-related acute liver injury? Is a ethambutol TDM a must?
Maybe I can quickly answer this one.
So
Yeah, actually, if there is a sign of hepatic toxicity, I think the rule of thumb is just stop on the hepatotoxic drugs like pyrazinamide, isoniazid rifampicin. Yes, stop on them first, and then evaluate like which one is more likely. Course decide. Yeah. And for the second question, actually, I'm not a pharmacist who practicing in TB. So based on my observation, most of the time, ethambutol is not a must in TDM. I think it's kind of an additional. It's not a key drug in treatment. Please correct me if I'm wrong, but my understanding is not a must.
Yeah, I'm happy to jump in with my thoughts on the liver toxicity side, and then we'll definitely get Jan-Willems on ethambutol PDM. But exactly right. We've gone back and forth a lot over the years, haven't we, on management of drug-induced hepatotoxicity related to TB. The issue is if we're on standard HRZE therapy, if you're only stopping the hepatotoxins, you end up with ethambutol as a single agent left over when we should never really be using single drug therapy for TB. So the traditional approach has always been to stop all drugs, exactly as T said, and then to reintroduce in a method that allows for the least hepatic drugs to be started first. Again, that's been hugely controversial. The IDFA guidelines for a long time recommended single drug week by week, and some people have been concerned about the development of resistance when single drugs are reintroduced. So there's a variety of approaches really now for the reintroduction. And yeah, I think still the party line is that all drugs would be stopped. And then, Jan-Willem, I'm hoping you've got an ethambutol PDM idea or?
Oh, yeah, yeah. Adding to that previous discussion, you can always consider when does the toxicity occur? Is it early during therapy, where there's still a lot of bacteria around, or is it more towards your continuation phase? So, the more bacteria, the bigger the chance that you acquire resistance on monotherapy compared to when there's only a few bacteria left lingering around. So for TDM of ethambutol, the only reason actually to do that is in case you have a renal function problem and you cannot drop ethambutol. That's really the only reason, because if you can drop it because of proven susceptibility to the other drugs, that would be the preferred choice rather than spending time and money on TDM of ethambutol. So dropping, if possible, is the first option. If not, then indeed TDM can guide your dose adjustments. Yeah.
So the next question is, could you please explain the therapeutic ranges, targets for anti-TB drugs?
It is a big one. I know we'll obviously want Jan-Willem's comments on this, and I guess maybe just to save his voice a bit, it is really always about having a reason for doing TDM. What's your rationale for doing it? It's still a fairly new concept, despite there being lots of not new, still fairly infrequent, I should say. It's not new. Yeah, maybe I'll just hand over to Jan-Willem. Any thoughts on which drugs and what targets?
Yeah, thank you. So, without giving a very extensive lecture on the individual drugs, because then we'll be here tomorrow, we will be publishing the TDM guideline for Australia and New Zealand soon, which has, based on available evidence, the best possible targets. And for some of these drugs, we have really clinically validated targets, whilst for the other drugs, we need to rely on in vitro PK/PD models to get a kind of target exposure range that we would like to aim for in case we start doing TDM. So the level of evidence for the different drugs is different. But basically, I mean, I agree with you. You should have a reason to do TDM in the first place. I always say TDM is not a goal. It's an aid to help optimize the care that you want to give to an individual patient. And if there's reasons like malabsorption due to critical illness or drug-drug interactions that you might expect the exposure might not be what you would like it to be, then there's a reason. So it's more common sense than hardcore science that guides our decisions in this case. Yeah.
I think we have Anton back with us, Amy.
I originally, when I heard about this meeting, I asked if I could just sit in and be a fly on the wall, because I just wanted to learn as much as I can from, people who actually do the research. I'm not a pharmacologist or a clinician myself, so I just wanted to learn as much as I can. And the journal that I'm an editor of, we actually publish a lot of papers on tuberculosis. I originally studied entomology, and then I ended up working at a medical research institution in the malaria field, where I studied the DNA and genetics of malaria mosquitoes. So I didn't enjoy that all that much, and I decided to join the pharmaceutical industry. Worked there for 10 years, where I learned all about pharmacology. And then I went into publication, where I'm now working for Springer Nature under the ADIS brand as the editor of the journal Clinical Pharmacokinetics. So I had a look over the last 10 years. I see that in my journal, we published 42 papers on tuberculosis, on the pharmacokinetics of drugs used to treat tuberculosis. And of the 42, 12 actually come from Sydney, from Jan-Willem's group. So that's almost a third, which is pretty good. Now, usually for these presentations, when I present to, especially to early career researchers, I have about 45 to 60 minutes where I can tell you all about the details of the publishing process and how to write up your paper and how to revise it and so on. But in the few minutes I have, I obviously can't go into all the details. So I thought I'll just give you some philosophical insights that I hope will be helpful. So I think what I came up with was, I think for an author to publish in a journal, there are three, I guess, characteristics that you would require. The first one would be realism. So you have to be realistic about the kind of journal that you want to publish in. The higher impact factor journals typically are obviously a lot harder. As a journal editor, I'm always looking for novelty. And I get a few thousand submissions every year, and I can only publish about 150. So even though your paper can be quite novel, it really has to compete with all those other papers. So you have to be realistic about the level of journal that you submit to. The second one is you need resilience, as I'm sure a lot of you know. You're going to get rejected, and sometimes you'll get rejected multiple times on the same paper before you find a home for it. But my advice is just to keep on at it. Just because you're rejected from one journal doesn't mean you're rejected from science in totality. It just means your paper doesn't fit that journal at this time, considering all the other submissions that the editor receives. So it's not a comment necessarily on the quality of your work. So just keep going with it. And then the third thing is you're going to need patience. As I'm sure a lot of you will know, it will take time to find the right journal. The peer review process can be very slow. It takes time. Peer reviewers do it, voluntary. So it's just the way it's going to be. So, realistic, resilience, and patience. Those are the three main things that you're going to need. And then just a quick mention to be very wary of predatory journals and predatory publishers who can often guarantee very quick turnaround times, like a week or 2 weeks. That's unrealistic. Once you publish in a predatory journal, you can't withdraw and then republish later. So just speak to your librarian if you're unsure about the journal. And then because we've got such a young audience, I thought I'd just briefly mention something about AI. They're all using AI now. AI obviously doesn't qualify as an author, so we shouldn't use it to generate text for our manuscripts, but you can use it for language editing. So that's totally fine. Use it to kind of brush up your language and your writing of your paper. But you can't use it to generate text. And we also don't allow generated images. The copyright issues come into it. And if you're ever asked to act as a peer reviewer, we also don't want you to enter any of that text into AI tool as a peer reviewer. So that's it's hard to police that, but that's what we request. But it's a changing field, and we'll obviously update our policies as time goes. So that's all from me. Once again, apologies that I just disappeared, but that's the way it goes, and I'm happy to take any questions.
That's great, Anton. I might just ask you a quick one and then we can get back into the Q&A. Is there anything that you see, or what are the things that you see that are direct reject? Just to give people a bit of idea.
Vast majority of papers so you talk about desk reject. That's when I look at it as an editor before I send it for review. And it's something I don't like it. So the desk rejects, the vast majority of them that I reject are very good quality papers. They're just not novel enough. So sometimes there's nothing you can do. That's just your work, right? It's just relatively, and maybe just go for a lower tier journal. I think in terms of, so that would be the main reason for rejection. It's just badly understand that for the majority of people, English is not your first language, and that includes me. So I understand that, and we have a certain level of tolerance for that. And if we accept your paper, we do copy edit it to fix it up nicely. But it has to be, go on the journal website and look at the format of the papers that are published there and write it up in a proper format. It just shows me as an editor that you've got attention to detail and you've gone to some effort just to dress your research up nicely, because it has to be, the peer reviewer also has to look at it and think, okay, it's a good quality paper, it's worth my time. But if it's sloppily put together and written and badly formatted, then it's usually a quick reject.
Thanks, Anton. We might get back into the Q&A. So the next question I might direct at Shukry, because it kind of tied into what you were talking about. So what interventions to improve pharmacist-led care exist for TB patients?
Yes. So as I mentioned, like interventions means like treatment or TB case finding or TB prevention. There are it's a broad area. So pharmacists can play in everywhere. As I said, mentioned earlier, in terms of adherence. So we had a pharmacy-led dot program, V dot program, direct observed treatment or therapy, and all those things. Pharmacists can play a huge role in terms of adherence to the TB treatment, and also TB prevention, as I said, especially TB high prevalence countries. It's obvious from some of our studies in Indonesia, Vietnam, and all. The patient with TB or presumptive TB symptoms, they first go to the community pharmacy for help. And then according to literature, up to 60% of these cases were not treated at the first point. So if pharmacists were well educated, they are able to identify the cases based on the symptoms. Yes, that is the key role they can play in order to identify in the cases early enough. And then now AI era, so AI tools can be incorporated in our research, like identifying the cases early enough through pharmacy or community level. That is also one thing. Then, yeah, these are the things I can capture at this time. Maybe he and others, if you have anything, please add. Oh, yeah.
Yeah, I might add on, an idea that I think, at least for my country, I think it's different, because the health system different from country to country. But at least in my country, I know that the healthcare system works like this. They start first with community pharmacy. Everything when they feel sick of they will go to community pharmacy is where we first detect TB. So I think community pharmacists in Vietnam play a major role in fighting cases. And then they go on, for higher level, centralized pharmacies, and then GP kind of and then hospital. So it's the way to work in Vietnam. So I totally agree with you, Shukry, on that. Yeah.
Thanks, Thi and Shukry. Did anyone else have any comments on that question? So the final question that we have received, and feel free to put any other questions that you may have in the chat. What are the strategies for linezolid renal dosing adjustment for advanced CKD patients with and without TDM?
But yeah, this is linezolid is another really interesting one where when it first came onto the market, because it had relatively low renal clearance, for a long time it was marketed as no dose adjustment was needed in patients with CKD. And obviously now we've realized that you do get very high exposures if you give standard dose in CKD. And we are also thinking now that you can probably head towards the lower end of the dose exposure range when you're treating for longer term. So essentially, in terms of dose reductions for patients with CKD, even I guess this is I'm a little bit more familiar with this in the NTM space than the TB space because we have such little MDR TB in Brisbane. But we do start with much lower doses of linezolid when it's going to be long term. So traditionally, even with normal renal function, for NTM, we would use 300 twice a day and then drop it to potentially 300 once a day. We are very lucky in Brisbane. We have pretty rapid access to linezolid TDM. We tend to do a trough concentration relatively quickly, maybe within the first week or so. And we, for, gram-positive infections, we have a range of 2 to 7, and we like to really be about 2 for our patients who are going to be on therapy for long durations, and particularly in the context of NTM and TB, where it's a multi-drug therapy. So to summarize all of that, and we'll get some more specifics, but essentially we use lower doses anyway, don't traditionally use more than about 600 a day, either 300 BD or 600 a day for the mycobacterial infections. And in CKD, you can often get away with very low doses, so potentially even just 300 once a day. What is really tricky with linezolid is a lot of the myelosuppression is dose dependent. And so particularly if you can do TDM, if you're seeing, myelosuppression, checking a concentration can mean that the drug can still be used with dose adjustment as opposed to getting a label of allergy, with the reaction being myelosuppression and then that drug being off the cards, where often we find really good bounce backs of the blood cells once the dose is reduced to, within the therapeutic range. But I'm very happy for you if Jan-Willem wants to add anything to that.
Thank you, Amy. So I think nowadays with assays becoming more readily available, so it's not only LCMS, but there's also immunoassays for linezolid. I think centers frequently prescribing these drugs should have access to an assay either within a reasonable a region that they can get timely results, or they should invest a little bit in their immunoassay and get it on their autoanalyzers. So that's one thing. The other thing is that in Netherlands, we also compounded a lot of linezolid capsules, particularly in that time era where MDR-TB was treated for 18 months with linezolid. So capsules made from tablets were often well tolerated for this very prolonged treatment. So as long as you do levels, you can go low, and in most situations, then tolerability is still happening. So you can make it work. Yeah.
Thank you. We have had one more question come through on the chat. Is TDM also helpful for linezolid related neuropathy?
Yeah, I would like to say yes, of course. Yeah. Just add on that we can do either drop or IUC monitoring, which is more and more meaningful and more reliable than the drop.
And so with your AUC, obviously with neuropathy, I think we have the cumulative AUC that can be predictive of peripheral neuropathy. And so would you maybe do a daily AUC and then work out how long it might take to get you there? Is that how you think about doing your AUCs for neuropathy?
I'm not too sure about that detail, but as far as I know, is AUC 24 for the therapeutic range. Yeah.
Oh, okay. Perfect.
So yeah.
What you would like to do ideally is prevent neuropathy from happening, because once it's already there, it's a big problem, and it can last for a very long time. So what we actually did, and this was a part of my PhD, I think over 2 decades ago, when linezolid was first introduced onto the market, we had our physiotherapist involved in monitoring these patients. So they tested from baseline on the vibration sense that patients have in their extremities. And once you see a change happening, that is a very early sign of neuropathy happening. And at that stage, you would consider, okay, what would be the most appropriate action for this patient? Reassess the levels. Can we go even lower? Or should we discontinue the linezolid before things get worse? Because I've seen patients ending up in a wheelchair as a result of this side effect. So it can be very severe and very long lasting.
Thanks, Jan-Willem. We have one more question. We probably have time for one more question. So this is from the chat. How can we pick the sampling time points and the number of samples per person necessary for TDM with saliva samples? T, do you want to comment on that one?
Sure, I think I'm the one. Have to. Luckily, we just published publication with Anton on that. So we published a paper about limited sampling strategy. So that shows you on possible effective strategy to go for saliva. So we found that even just one saliva sample at drop, so pre-dose one, can reliably predict the AUC of linezolid and also levofloxacin. And therefore, of course, that the more the better for the accuracy. So you can do like 1 to 2 to 3, up to 3. Within 12 hours after dose. That will give you a good estimate of AUC.
So those ones taken within the first 12 hours after dosing, is that what you're saying?
Yeah. Yeah, you can search saliva sampling strategy for linezolid and levofloxacin, and that should pop up with the CPKs, and you can read the full text for more details.
All right, well, it is getting up to 2 o'clock now. So I'd just like to thank Shukry, T, and Anton for speaking today. I hope that this session has provided some insight into the important research underway in the TB space, and also given some helpful perspectives on how people can become involved in this really very interesting area of research. Also, just an announcement about the next ACTnet event. It's the upcoming Journal Club with Dr. Emma Smith on Tuesday, the 16th of June at 1 p.m. And we'll be presenting a paper titled Weekly 3HP and Daily 4-RIF for TB Infection, a randomized trial. Nishida will add the registration link in the chat for anyone who would like to attend. Thank you, everyone, for taking the time out of your busy days to attend. And we'll see you next time.